The following program is a public access production of Ward O'Hara Agricultural and Country Living Museum, who assumes full responsibility for its content. The content of this program does not represent the views of Auburn Regional Media Access, which serves as a forum for community expression and offers TV training, production facilities, and channel time to all Auburn area residents and organizations.
Thank you guys for coming out this evening. My name is Frank Clarke. I'm with Cornell Cooperative Extension here in Cayuga County. The office right in Auburn on Grant Ave. It's our new office. It's our old new office because we just renovated it. We moved back in about a year ago. So I'm here to talk about some research that's tracking like the history of Cyanobacteria in all of the Fingerlakes. They did that using very deep core sampling methods. So that's what we're going to get into. But before we start, I should note, and he alluded to this, the bulk of this research was done by everyone on the right of the screen there. And Doug Young was like spearheading this operation. He was using his boat to take people out to take those cores. So this is their research. I was a part of a different set of research that looked at the cores, which I will present on, but the cyanobacteria stuff that we're going to talk about, I did not do that research, so I'm going to do my very best to portray that information as best as I can. I'm a soil scientist by training, not a algae expert, or a lake expert, so I'm going to do my best, but bear with me a little bit. Okay. So to set the stage here, we're going to do four things. We're going to learn about algae and cyanobacteria, how they differ. We're going to learn about what makes harmful algae blooms harmful. And then we are going to talk about why HABs in the Finger Lakes is kind of puzzling. And then we'll go over the results and what that tells us. You're not getting any answers to why we have HABs, unfortunately, from this research, but this does inform us quite a bit and gives us some interesting information. Okay, so there are algae and then there are cyanobacteria. Algae are complex cells. They have nucleus. They photosynthesize. They are pretty much small plants. They are plants. Whereas cyanobacteria, which causes harmful algae blooms, are not actually algae. They are a bacteria that also photosynthesizes, but they contain a pigment called phycocyanin. That's probably pronounced wrong, but it has an extra pigment, unlike algae, that gives them that blue-green color. It's like a blue pigment, hence the cyan in the name. So that's the microcystin, so that's what causes HABs, and then those three other names that I'm not going to try to pronounce. But the thing is, they are not algae, so when we say harmful algal blooms, it's really caused by cyanobacteria, but we'll still call them algae blooms, but just a little caveat. Okay, so what makes a harmful algae bloom harmful? It should be noted, most algae blooms are not harmful. They are natural occurring events. Most lakes that aren't super clean get algae blooms. It's just a natural process in lakes normally. So what makes a HAB harmful is the release of microcystin, which is a liver toxin that can damage the liver, kidney, and can have some pretty nasty effects. That's the most common one. That's the one we are most nervous about. There are those two others. They can be pretty dangerous, but we don't see those very often in the Finger Lakes. There are EPA drinking water standards. They are less than 1.6 micrograms per liter, which is fairly low. We'd prefer no microcystin in our water, which is usually how it happens. But some microcystin isn't going to be too dangerous, but we still don't want it in our water. Okay, so this is kind of the classic mindset of what causes an algae bloom is warm water. So the cyanobacteria operate best when water temperature around 70 degrees Fahrenheit. They like lots of phosphorus. In most freshwater lakes, the limiting nutrient is going to be phosphorus. There's usually more than enough nitrogen and other micronutrients to support algae. It's usually phosphorus that holds them back. So when there's a big flush of phosphorus and the water temps are correct, they can usually get that big bloom. But usually you need calm water. They don't like waves. They don't like being agitated. If you agitate an algae bloom, you can actually stop it. You have to do that at a huge scale, which is why we don't do that as a method. And then you want shallow, productive lakes. By productive, we just mean high in nutrients. There's a lot going on there. They're active lakes. Lake Erie is probably our most famous nearby example of a shallow, productive lake. So we want warm water, lots of phosphorus, calm water, and productive, shallow lakes, which the Finger Lakes are not those things. The Finger Lakes are typically pretty cool. They are ultra clean lakes. They don't stratify quite a bit. They are calm, but they aren't overly productive lakes. So that is why having HABs in our lakes is so confusing or puzzling to a lot of scientists, because most of our lakes are oligotrophic. So there's eutrophic, which means high in nutrients. That's where you see like those muddy green lakes. That's a eutrophic lake. There's mesotrophics, that's that middle ground. And then there's oligotrophic, which is very little nutrients. So which ones? Honeoye is a eutrophic lake. Skinny Atlas is oligotrophic. Owasco is like meso-oligotrophic, so it just hovers between that line of being ultra low nutrients to that middle ground. Some years it's oligotrophic, some years it's mesotrophic. So for all intensive purposes, it is a low nutrient lake, should not be having HABs. Quote from the president of the Skinny Atlas Lake Association in 2028, we thought Skinny Atlas Lake was immune to HABs. Because it is so clean, it is a deep lake, HABs shouldn't be showing up based on our past framework. And it's very important to keep Skinny Atlas clean because it is a unfiltered drinking water source. It's one of 4 unfiltered drinking water sources in the entire United States. Skinny Atlas Lake, New York City's watershed, and then I believe there's one in Massachusetts, and then another one in Washington. So it's really rare to have an approved unfiltered drinking water source. So that's why, and then all the other Finger Lakes are drinking water sources. They are filtered and treated, but that's why we are, nervous about having HABs. And this isn't a Finger Lakes specific problem. Blooms in low nutrient lakes have been popping up pretty much everywhere. You can go to some of the more pristine lakes in like the Adirondacks, and they've seen HABs and they have no human influence in them. So this is a new problem that has confused people for quite a long time. Okay. So that's kind of the big picture. We're going to hone in on the Finger Lakes now. So this is just kind of a small timeline of HABs in the Finger Lakes. And then I threw in the Toledo example just because that was a major news thing. They had to shut down drinking water for 500,000 people. So it's pretty huge. So I threw that in there. That happened in 2014. 2016, Owasco Lake has a microcystin scare, so that they detected that toxin in the drinking water. 2017, Skinny Atlas gets its first confirmed HAB, so a harmful algal bloom with those harmful cyanobacteria. Canandaigua has some issues in 2018. 2021, Cayuga Lake has high toxins in their lake. And then 2024, just a couple of years ago, we have record-setting HABs in all of our Finger Lakes. So this is a relatively new issue. We haven't had HABs in the past. But obviously, monitoring has gotten better, so that does adjust for some of like the increased numbers we're seeing, but it doesn't tell the whole story. So we're trying to figure out why this is happening. So we have 2 kind of hypotheses. The cyanobacteria are new to these lakes. They invaded our lake somehow, or they've been in the lakes this whole time, but something's changed that has, made their population spike and given them the ability to cause harmful algae blooms. So either they're new, kind of like an invasive species, or B, that they've been around for a long time, but they just kind of have had low populations. Okay, so we're going to jump into the research now. So they sampled 6 Finger Lakes, Honeoid to Skinny Atlas. Cayuga has a asterisk next to it because those samples have not been ran yet, so we don't have the data for that. But we have a pretty expansive data set to gather quite a bit of information. So like I was saying before, Honeoid eutrophic, really high nutrients. Owasco Lake, mesotrophic. Canandaigua, oligotrophic, really low nutrients, really deep, 130 meters. Cayuga Lake is probably more mesotrophic at this point. And then Seneca and Skinny Atlas, both oligotrophic, 120 plus meters, really deep lakes, not what HABs are supposed to have, which are shallow lakes. Okay. So the research they did is kind of like a time machine. Essentially, what they did is they take the 3-meter cores, so really large cores, and drive them through the sediment, and then they pull them up, and we can use those cores we can date them with lead dating or radio dating, and we can get a bit of a time machine based on those slices of sediment they used. So they took, what did they do? Okay, they sliced it every ½ centimeter. So there's a lot of information coming from that. So they took these very deep cores and then sliced them just into ½ centimeter sections. And each of those ½ centimeter sections was like a data point that they could look into. So a lot of stuff going on here. Using lead-210, which is a natural radioactive isotope, it decays at a known rate. I believe its half-life is 22 years. So essentially, depending on how much of this lead isotope is in that ½ centimeter slice, we can tell how old it is because of how quickly it's decayed. We know how much should be there based on, because usually that lead isotope does fall from the atmosphere naturally, and that's how we're able to date it. And then in each slice, we measured for pigments of algae, DNA from cyanobacteria, and toxin molecules that are all preserved in the lake's bed sediment. And we were also able to cross-check our dates to confirm that our dating was correct for each of those slices, because Seneca Lake and Cayuga Lake had a coal-burning power plant nearby the lake, and burning coal releases mercury. So we were able to kind of date here with the increase in mercury while the plant was active, and then when the plant was shut down in the 2010s, that number fell. So we can cross-check that with our lead dating, with this mercury dating based on when we saw those power plants, and they line up pretty much exact, which is confirms that we're pretty spot on with our dating. Okay, so what did they measure? So we measured chemistry and genetics. Both are chemistry, but we're going to split them apart. Total chlorophyll, so the pigments from all the algae and cyanobacteria that they use to photosynthesize. That tells us how much algae was there based on the amount of pigment there was. That cyan pigment that's unique to just cyanobacteria. So if we see a lot of that cyan, we know it came from that specific cyanobacteria. And then it should be known that pigments do degrade over time, so some of the older layers may underestimate how much was actually there. And then we did look at the genetics of cyanobacteria and some microcystin-producing genes. So we were able to monitor that as well. It's important to know, just because the microcystin gene is there does not mean it was active. So that does not mean toxins were being produced. It just means that those cyanobacteria had that genetic code in them. Just doesn't mean they were using it all the time. Like an example is if you found a recipe card in someone's kitchen, someone's capable of cooking that cake, but just because you find a recipe card doesn't mean they actually made it, if that makes sense. Okay, so now we're going to get into the results. Okay. This graph is confusing to me as well, but this is what we found. So this is total chlorophyll over time. So our y-axis up here is the calendar year that we dated it for, and the x-axis is the amount of chlorophyll pigment we found in the sediment. So essentially, the big picture is there's a shift around the 1960s where we're seeing a increase in algae over time. There's, for some reason, a very big spike there. Honeoye Lake, it's a eutrophic lake, so it is elevated. That makes sense to us. The low numbers make sense to us in ultra clean lakes, but something did happen around here that caused a large spike. So that's just something interesting. Looking into the history, we don't know exactly why that happened, but this chart here is a zoom in from 1995 to 2025. And the nice thing about this graph is we can actually see the lakes are lowering in algae concentrations. We can't, say for sure why that is happening. Maybe it's reduction in erosion, more nutrient control. We don't know exactly why. But this is all algae, not just cyanobacteria, the HAB causing algae. This is all. Okay. This is chlorophyll flux over time, whereas the last one was concentration. So we're going to try to make this a little clear. So concentration, so let's say we went to the store, we bought apples. How many apples are in what percentage of the apples in the bag? That's the concentration of apples. The flux, what we're looking at now, is just how many we actually found. So we do see an upward trend as well in the flux. So when the flux and the concentration, align, something is happening. There is an increase in algae. So why flux matters is we can have a huge storm, one of those 100-year storms that dumps a ton of sediment or clay into the lake. That would dilute the amount of algae in there. There's a lot of clay to algae ratio, but that doesn't mean there wasn't a lot of algae. That just means a lot of sediment fell into the lake. So we see an increase in concentration of algae, and we see an increase in the flux of algae over time. So those line up, which tell us there's probably something happening. There's a reason why algae concentrations are increasing. Okay, so if we zoom in on cyanobacteria, this is our HAB-causing bacteria. This is the one we care about. This is what's causing the issues. This is where we see probably the most interesting data. So this is now different. This is a Z-score. You don't need to know what a Z-score is. 0 means average. -2 means below average. +2, above average. +4, really above average. 6, an outlier. So we see a jump in cyanobacteria starting in the 2010s, which lines up with when we started to see HABs in the Finger Lakes, which is odd. Why did they all of a sudden start reproducing at such high rates? We're seeing a huge concentration, a big change in cyanobacteria populations over time when nutrient levels in the lakes haven't really changed. In about 50, 60 years, the nutrient levels in most lakes, including Owasco, have remained fairly steady. So it's not like there was this huge jump in phosphorus concentrations in the lake that could explain why we have such a huge jump in cyanobacteria. So it is kind of puzzling why that's happening. And that's kind of what motivated this research was, why is cyanobacteria showing up it should have, nothing's changed to introduce cyanobacteria. Nutrient levels haven't changed. Farming practices have gotten a lot better. They've reduced the amount of nutrients that are coming into the lake. So it is a bit perplexing that we're seeing this big jump. And one of the explanations that some people hypothesize is the introduction of the zebra mussels or the quagga mussels. That introduction does line up roughly with when we started to see this change in cyanobacteria. Okay, so this is essentially just to confirm what we saw in that past research. This is to look at cyanobacteria DNA. And again, we see that cyanobacteria DNA is in the lake. It's existed in the lake for the past 100 years. But again, for some reason, in about the 90s, we saw a large increase in that cyanobacteria DNA, which again doesn't exactly line up when we started to see the HABs, but something was happening around here that caused cyanobacteria to increase. There is some variability with the lakes, but the trend we think is real. There is a real change going on. So this is looking at the microcystin gene. So this is the gene the cyanobacteria have to have in order to cause the harmful part of the algae bloom. They need this gene to produce microcystin. That is that toxin that hurts us if we drink it. And again, we just see a spike in this DNA, this genetic code that can cause microcystin around the 2010s, even though we did have it in the lake for quite a while. But for some reason, it's spiking in the 2010s, about when we start to see our HABs. In the Fingerlakes, caveat, DNA degrades over time. So the later dates, so like the 1920s to 1960s, those could be underestimating the amount of that DNA that's in the sediment. But we still think this trend is actually happening. We think this is a real trend because it does kind of line up with all the other data sets we've looked at where there's just this odd increase in microcystin, algae, cyanobacteria. So these things are all lining up, kind of telling a very similar story. Okay, so what does this tell us? Cyanobacteria is not new to the Finger Lakes. They've been here for 150 years. That's how far back we've gone. Both pigments, the DNA increased over the last several decades, even in the skinny atlases, which are deep, cold, low phosphorus lakes. They are, really low. They shouldn't have harmful algae blooms. The genetic capability of those cyanobacteria to produce mycotoxins was always in the lake, but again, there's a recent increase we've seen in the sediment over the past couple decades. The why on this is happening is still very open. This was more of like a history of the lake. We weren't trying to answer exactly why we see those spikes. This was mainly to look at, is there a change or is the history the same? And the history shows us that the cyanobacteria were here, but for some reason they're spiking. The gene copies, that's the ability, that was the last slide. Again, we're seeing it increase. That doesn't mean the toxin is being produced. That just means the ability for them to produce the toxin is increasing. We don't know exactly why these things are happening. We can hypothesize that climate change, so warmer waters, could be a cause. Nutrient loading, although nutrient loading has decreased over the past couple decades. Invasive mussels, atmospheric deposition of nitrogen, which has gone up, or something completely different that we don't know. But those are just our best guesses as to what could be causing these changes, but we don't know exactly why. We just know we're not going crazy, and there is this weird shift that happened about, 10 to 20 years ago. So now we're going to move on to the soil or the sediment. One thing we should know, We used a agronomic soil test to do this. So this is a soil test you would use for your farm field or for your home garden. The reason why we did this is because it costs $15 to do a sample, whereas to do a in-lab at a university would be hundreds of dollars per sample. It's like usually like $100 per nutrient. It's wildly expensive, and that's just not something that was in that research that they had the budget to do. So that's why we did this agronomic method. This is just going to give us an idea. This isn't the end all be all, because this is an agronomic soil test, which doesn't measure exact amount of nutrients. It measures usually what's plant available. So there is some differences between a total nutrient test and then an agronomic test. An agronomic test isn't pulling all the amount of phosphorus that's in the sediment. It's just looking at it like a ratio. Okay, so we took 5 cores. They are all from Cayuga Lake, 0 to 5 inches, 5 to 10, 10 to 15, and so on to 30 inches. And then we just did the modified Morgan test, which is a normal soil test. It's not the right tools to use for this, but it does give us a pattern, a relative pattern, just something to think about. My hypothesis going into this is that there would be high phosphorus levels like towards the surface, and the reason that would be there is because of organic matter, so they would correlate fairly well. That's what I was thinking, because of runoff, erosion from, that usually pulls off organic matter off the field. So that was my hypothesis there. But if we look at what we measured, we actually see, organic matter is decreasing with depth. So as we go deeper in the cores, organic matter decreases. That makes sense. Some of it gets decomposed. There's not new organic matter being added on, so we would expect it to decrease with depth. Phosphorus, again, we would expect to see it drop down a little bit as phosphorus binds with other minerals and it does decompose or gets used. And then we would expect to see iron increase as you go into depth because it is anoxic, so iron can settle out, and that makes sense to happen. The weird one was probably calcium, because we should expect to see that same just decrease like we saw with organic matter or phosphorus. What we see is pretty high plateau here and then a decrease. That is a bit of an interesting result. So if we run a model, the model suggests that iron and calcium controls phosphorus in the sediment. So if you have a lot of iron, you'd expect to see a lot of phosphorus because it binds it together. And the reason why we did this, our hope, not hope, but what we were thinking about what we might see was the opposite of this phosphorus graph. We were thinking, okay, if farming practices were not great 60 years ago because we just didn't know, and there was a lot of plowing, manure applications, higher rates than they needed to be, we thought, okay, maybe there'll be a sink of phosphorus deeper in the core. That's what we were kind of expecting to see. We didn't see that. We just saw a normal trend. So we were, the idea was, well, maybe it's the phosphorus that's sitting at the top of the sediment that may be feeding the cyanobacteria, and if that is happening, what is the reason phosphorus levels are elevated? We thought it would be organic matter, but really it was controlled by calcium and iron. And this model just kind of corroborates that. Most of the variation we see of phosphorus is controlled by iron and calcium. But again, our sample size was fairly low. This was 5 cores going down 6 different depths. This is a fairly low sample size, so there's no significance that I would take from this. Okay. This is now my theory here, is if we go back to this calcium level, quagga mussels, zebra mussels, they are largely composed of calcium carbonate. When they decompose, they release a lot of calcium because they are made of calcium. If calcium is controlling phosphorus, it's possible that the calcium from quagga mussels is holding on to phosphorus, and that is able to be released, whereas if it's held on by iron, it's pretty steadily bound. So that's just why we found it interesting to see that like plateau of calcium at the 0 to 5, 5 to 10 depth, is because maybe that has something to do with increasing in calcium from the mussels. To confirm that, we'll have to do another set of core samples where there are no mussels. So these were done in a mussel zone. There are certain patches around the lake that contain higher levels of mussels. That's where these samples were taken. The next step would be to take samples again in those mussel zones, but then in another zone that does not contain any mussels. And if we see the same pattern, then it's probably just a natural phenomenon that the calcium is high at the surface. If we see calcium levels low in the no mussel zones, then maybe something's going on. But again, this is like the past research, this is preliminary. We're just trying to see some things. So there's no silver bullet solving HABs here. So what's coming next? They are trying to measure the actual toxin rather than just the gene. They're going to see if they're able to actually see when toxins were produced rather than not just the recipe to make toxin. They want to see the actual toxin. They are also going to do phosphorus, nitrogen, and carbon testing on those samples that was on the first part of the research, but that's taking quite a while. And then they will also do an analysis on the Cayuga Lake course, which were not included in this. I think it's just important to know just about everybody cares about the lakes here, whether you're a scientist, homeowner, fisherman, boater, or just drink the water. We all care about the lake. That's why we're all doing research. We're all trying our best to figure out what's happening and what we can do to kind of hopefully prevent HABs in the near future. So that's what I got. I hope you learned something. It's a lot of information, I know.